Now, switching gears, homecoming is a chance to come back to campus. And as you see, I'm standing virtually in front of McGaw Hall. So shout out to all of our BioSci alumni. I want to introduce an amazing presenter we're lucky to have today, a professor in the School of Biological Sciences, Aimee Edinger, to talk about her research in using cell biology to treat obesity. Aimee.
Okay, let me share my screen so we can get going here. Okay, thank you. I'm so happy to participate in homecoming this year and to have an opportunity to tell you a little bit about our recent research. I do need to start with a disclosure. So I started a company, Siege Pharmaceuticals, to develop the small molecule that I'm going to be talking about today. And if you're interested in that process, I recently recorded a podcast for the UCI Beal Applied Innovation Rising Tide Changemaker series. So you can check that out if you want to hear more about the company. Okay, so I think probably everybody in the audience is aware that obesity is a problem, but you may not realize quite how serious that problem is. The CDC estimates the annual cost of obesity, the medical cost, is $147 billion. Because of its lethal comorbidities like cardiovascular disease, fatty liver disease, and type 2 diabetes, obesity is actually the second leading cause of death in the United States. And the recent article in the New England Journal of Medicine estimates that 4 million people die each year because of obesity. So clearly that puts it on par with COVID-19, the COVID-19 epidemic. So clearly obesity is a problem that we need to solve. And one of the obstacles that we have to overcome in treating obesity is this resistance to the idea that it's a disease that requires medical therapy. For decades, the general public and even physicians have felt that obesity is the result of a lack of willpower, and they've prescribed eating less and exercising more. Well, clearly this treatment plan has been a failure. And I'll say the science tells us that obesity is not the result of a character flaw. Instead, the fault lies in our genes and in how our body responds to life in the modern world. I think that the television show, the reality show, The Biggest Loser, provides a really useful illustration of why it's hard to lose weight. So in this show, contestants were supervised by dietitians and personal trainers, and some of them were able to lose spectacular amounts of weight, such as Danny Cahill, the winner in the year I'm going to talk about. You can see her on the right. The graph shows he was the biggest loser. But unfortunately, despite the fact that these individuals continued to exercise and carefully watched what they ate, 13 out of the 14 participants regained a lot of the weight that they lost. And one of the reasons this happened was because of changes in their metabolism. Their bodies burned as much as 800 fewer calories than would be expected based on their body weight after they'd lost weight on the show. And even years after the show ended, contestants described extreme hunger and constant food cravings. And this makes it really difficult to maintain a metabolic equilibrium. So this is a great illustration of the fact that our physiology can alter with obesity. And in order to treat this disease effectively, we need to understand the changes in cell biology that underlie and drive this disease. So it is a complicated disease, but I'm going to talk to you today about one biological change that people agree is at the root of obesity, changes in the shape of an organelle called the mitochondrion. Now, some of you may remember from biology classes in school or in college, this cartoon of a cell showing all the different organelles. And these orange bean-shaped structures here, these are the mitochondria, also known as the powerhouse of the cell. Some others of you might be more familiar with this image if you have kids. So this is a cell cake. It's an extra credit project often given to elementary school students. And if you Google cell cake, you'll see pretty much every student chooses Mike and Ike's or jelly beans to represent the mitochondria. It is clearly dogma that mitochondria are beans. And so one of my favorite moments as a professor is when I get to rock my students' world by revealing the truth that mitochondria are actually shaped like spaghetti, not beans. So in this image of a cell, the blue circle here is the nucleus where the cell stores its DNA. And these green tubules out here in the cytosol are the mitochondria. Now, in different cells, mitochondria can have different shapes, but this tubular form is really the healthiest, happiest form of a mitochondrion. Now, interestingly, when people or animals are on a high-fat diet and become obese, these mitochondria change from spaghetti into a more bean-like form, and this has actually been shown to drive obesity. So we can actually genetically engineer mice to have mitochondria that are always in this round, fragmented form. And when you do that, the mouse, even when it eats a healthy diet, becomes obese. Now, on the other hand, you can also engineer mice to have mitochondria that always look spaghetti-like, that have really tubular mitochondria. Now, these mice can eat an unhealthy, fatty diet, and they remain thin. So together, these experiments show that changing the shape of mitochondria is sufficient to either cause or correct obesity. And this suggests that mitochondrial shape might actually be a good target for drugs that could treat human obesity. Okay, so to monitor how mitochondria shape changes in response to a high-fat diet, we can actually mimic eating fat in a cell culture system. So here I'm showing you a cell. This is the nucleus, and the tubules in white are the mitochondria blown up here at the bottom. They look very spaghetti-like when we feed the cells a healthy diet, like this lunch shown here. Now we can mimic feeding the cells a bacon cheeseburger by supplementing their media with the fat palmitate. And when we do that, you can see the mitochondria end up in this fragmented ball-like form, just like they do in an animal. So we engineered this drug, which we call SHBC 893. And if you treat the cells with 893, you can feed them a cheeseburger, but their mitochondria stay tubules. And in other experiments, we showed that keeping mitochondria in this tubular form preserved the ATP content in the cell. So ATP is the fuel that cells burn to do their job. It also prevented the levels of toxic metabolites that are associated with aging and cancer, as well as obesity, from climbing in these cells. So that was nice that we saw this work in cell culture systems, but we wanted to see whether we could protect animals from obesity. And so we used a very common mouse model. A particular strain of mice is fed either the standard healthy chow or a high-fat diet where 45% of the calories are coming from fat. And after a couple months on this diet, mice eating the high-fat version end up about twice as heavy as mice eating the healthy chow. This is considered a really good model for human obesity for a few reasons. Number one, obesity is caused by a lot of different genes. And in this model system, there are lots of different genes that are contributing to the obesity. We're feeding them a pretty physiological diet. This is a good match to what humans might actually eat when they become obese. The weight gain is gradual. And the mice also develop the same comorbidities that people get. They get cardiovascular disease, fatty liver disease, and type 2 diabetes. So we took mice, maintained them on the healthy chow diet or a high-fat diet, and then treated some of the high-fat diet mice with our small molecule 893 and looked at the shape of the mitochondria in their livers. Now here I'm showing you what mitochondria look like in the livers of mice eating a healthy diet. This is in this box here is one liver cell or hepatocyte. This black circle here is where the nucleus would be. And all these white sort of hot dog shaped structures are the mitochondria. Now they don't look as nice and long as spaghetti-like as those cells that I showed you in tissue culture, but you can clearly see they change their shape when you feed the animals a high-fat diet. They go from these hot dogs into a more round meatball-like form. Now, what was really exciting is if you take these mice on a high-fat diet and you give them an oral dose of 893, just 4 hours later, their mitochondria now look just like the mice eating a healthy diet. Because I think this is so exciting, I wanted to show it to you extra large. So here on the left are mitochondria in mice eating a high-fat diet treated with water as a control. And on the right, this is how they look when we give them the small molecule. So it took those round, nasty mitochondria and made them tubular. Now, in that Biggest Loser example that I was talking about, hunger was one of the reasons why it's so difficult to lose weight and then keep it off. And that's because obesity actually damages the circuits in the brain that control appetite. There's been genetic studies in mice showing that the shape of the mitochondria actually controls appetite. So here is a mouse brain, sort of cut like a salami, and I'm showing you mitochondria in a portion of the cortex of the brain. And these mitochondria look kind of like the liver. They're tubular. Now, when you feed mice a high-fat diet for a couple of months, those mitochondria again become round. But if you treat the mice with our small molecule 893, you can restore those mitochondria back to the normal shape. And this image was taken just 4 hours after a treatment with the drug. And I'm going to pop a graph up here just to show you that we do use image analysis software in order to quantitate these changes in mitochondrial shape. So you can see that the mitochondria become more round on the high-fat diet, and that change is corrected by 893. Okay, so it turns out that the brain listens to signals from fat to tell you whether or not you're hungry. So that may kind of surprise you that your fat plays an important role in controlling appetite, but it does. So I'm going to show you the shape of mitochondria in fats, or the functional state of mitochondria in fat cells. So I want to show you what fat tissue look like so you would be able to understand those images. So this is what white fat looks like under the microscope. Here, there's some muscle, this kind of pink river here. There's a blood vessel down here. And most of these big round balloon-shaped structures are actually the fat cells. So I'm going to look at, let's see, where's a good one with here? This one's a good one. So there's a thin rim of cytosol that's pink around this big white empty space. And this white empty space is a big lipid droplet filled with fat. This little purple dot here is the nucleus of the fat cell, and these purple dots in between are actually inflammatory cells called macrophages. All right, so now we'll go to the mice that we treated. So the first thing we did, because the fat cell has so little cytosol, it's really hard to look at the shape of the mitochondria. So we looked at the function instead. And to do this, we use this special dye called TMRE. This fluoresces in proportion to the cell's energy charge. So it's sort of like applying a battery tester to the mitochondria. When you see red fluorescence, the mitochondria is charged. So here are these rings of cytosol around the lipid droplet in the fat, and you can see the mitochondria have a charge normally. But when you maintain mice on a high-fat diet and treat them with water, you hardly see any signal from the mitochondria in the cytosol. Basically, there's a dead battery in the fat cells. If you treat with our small molecule, however, you once again restore that energy charge in the fat cell mitochondria. We also used another dye to look at this toxic chemical called reactive oxygen species. In mice eating a normal diet, there's not a lot of these toxic molecules, so you can't see the outline of the fat cells. But in animals fed a high-fat diet, they make lots of these toxic chemicals, and now you can start to see a signal from the cytosol. If you treat with our small molecule, that signal goes away, so the toxic molecules are gone. Now, it was really nice that we restored mitochondrial function in fat, but what did that do for the animals? It turned out it normalized blood levels of hormones that the fat makes that suppress appetite and promote metabolic health. So fixing the mitochondria in the fat made the fat start functioning normally and send the right signals to the brain about appetite. Okay, so now we're actually going to look at weight loss in response to this small molecule. So we had animals that we treated on Monday, Wednesday, and Friday with the compound 3 days a week. And what we saw was that it caused rapid weight loss. So here in red are animals on a high-fat diet that were treated with water, and you can see they continue to gain weight. While in blue, the animals that were also given the compound lost weight, even though they continued to eat the high-fat diet. Now, for reference, an obesity drug is considered to be effective if it causes 5% weight loss. So this is clearly meeting that threshold for effectiveness. Now, these mice are not losing weight because they're sick. When we gave them a running wheel in their cage, they ran 2 kilometers a day, free choice, whether they had the drug or were given water. So they felt good enough to run 2 kilometers each day. And it was also exciting that allowing them to exercise further enhanced weight loss. So that's here in green. When they're allowed to exercise as much as they want and they receive the drug, they even lose more weight. This is over the course of just 3 weeks. Okay, so that weight loss is exciting, but the reason why obesity kills is because of the metabolic disease that it causes. And so we also wanted to look at how 893 affected the metabolic state of these mice. And we found that it corrects some nasty comorbidities. So it got rid of these lipid droplets in the liver that you see in mice on a high-fat diet. These are very dangerous. After treatment with the compound, those droplets go away. And it also corrected diabetes in these mice. Now, if you've been if you're pregnant and or have been pregnant, you're in the audience, you've probably taken an oral glucose tolerance test like we gave to these mice. So here at the beginning of the experiment, we give them a glucose solution. And so that's sort of like the nasty orange soda you have to drink when you're pregnant. Then what we do is we monitor the glucose in their blood over time. And in white here are the normal mice. So you give them the glucose solution and the blood glucose rises, then insulin gets released from the pancreas into the blood, and then the tissues start absorbing that glucose. So that's the normal response. Animals on a high-fat diet, they get the same glucose solution, but their blood glucose skyrockets. It goes up way too high, and it takes a long time to come back down. Now, in blue, we have animals eating a high-fat diet treated with the small molecule, and you can see now their blood glucose goes up and down, just like the normal healthy chow mice. So that was very exciting. All right, so I'm going to finish up here by summarizing what I've told you with a model figure. When animals or people consume a high-fat diet, their mitochondria become fragmented and bean-like, and this results in them losing their energy charge and starting to make toxic molecules called ROS that make obesity worse, and they also can lead to aging and cancer. Now, these changes in mitochondrial biology make the fat cells sick, and they reduce the amount of a good hormone. They secrete and over-secrete another hormone that controls appetite. This causes the brain to stop listening to what the fat is saying. The messed up mitochondria in the brain disrupt the circuits that normally reduce appetite. So what happens is mice that are fed a high-fat diet, fragmented mitochondria, they're hungry all the time, they gain weight, and they exhibit metabolic dysfunction. Now, if you can make the mitochondria stay tubular, then you can restore their energy charge and get rid of these nasty, damaging ROS. This makes fat function normally, so it makes hormones that promote metabolic health and decrease your appetite. The brain listens to those signals, and as a result, the mice eating the high-fat diet have reduced appetite, they lose weight, and they're metabolically much more healthy. I didn't really have time to explain today how we designed this small molecule 893, but I'll tell you, it's a drug-like version of a natural compound called Phytosphingosine. And Phytosphingosine is made by yeast when they're under stress. If you put Phytosphingosine in the culture, you can also stop the mitochondria from becoming rounded up when you feed them the cheeseburger equivalent. And so this is highly speculative, but maybe eating dietary Phytosphingosine, foods that have a lot of yeast that have been stressed, that might actually have metabolic benefits through a similar mechanism to what I described here. Okay, so I'm going to conclude by thanking the people who did the work. On the left here, I'm showing you a former graduate student, Lisa Selwan, who is a very brave person. She brought work on obesity to my cancer lab and really started these studies, did a ton of mouse work. And on the right here is Vaishali Jayashankar. She's a former postdoc in my lab, who was an expert in mitochondrial biology and took a lot of those beautiful pictures that I showed you. Now, I want to emphasize this is highly collaborative project. Most good science is the result of teamwork, and people as far away as Australia performed really important part in this study. And I want to particularly call out in Montreal, my chemistry collaborators, who's the SH, Stephen Hanessian, and BC of SHBC 893. These are the chemists who initially made the compound. And then there were a bunch of people here at UCI who provided expertise, reagents, or participated in experiments that made this work possible. And so with that, I'm going to stop sharing my screen, and I'd be happy to take any general or specific questions from the audience if you want to talk about anything.
Wonderful. Thank you so much, Aimee. We have a couple questions here. So first off, do you see the conversion of mitochondrial shape when there is weight gain from medications, like insulin or steroids, in spite of a healthy diet and exercise?
Yeah, that's a great question. So we looked at a really specific model here where we fed the mice excess fat and we saw the mitochondria turn into balls. There's indications from the literature that lots of different things that make people obese will actually cause similar changes in the mitochondria, but that's something we hope to look at in the future. I think there's good evidence that high sugar diets, so the fructose, the high fructose corn syrup in a lot of our foods can do the same thing to mitochondria. And when it's together with fat, it makes things even worse.
All right, let's see. There was also a question about the name of your company, and then perhaps you could share just a few words about the work that you're doing there.
Sure. Yeah. So it's called Siege Pharmaceuticals because when we first started working on these compounds, we found that they take nutrient transporters on the cell surface and downregulate them. So it's sort of like sieging the castle, right? This was originally a cancer therapeutic. And so the idea was we would stop the cells from getting access to nutrients, just like you do when you're sieging the castle. So that's where the name came from. And, you know, I think the issue is people may not realize that you cannot develop a drug in academics. It's just not possible. So the kind of experiments that you need to do are super expensive and they're not hypothesis driven. And so what motivated me to start the company was that if I wanted to test whether our ideas and research could actually benefit human health, I was going to have to start a company. And so it's been kind of a scary thing and it's been a good adventure and I've learned a lot from it. So if you want to know more, you should go check out the podcast. And actually, Olivia, I can see the Q&A too. There's a question, did the high-fat diet mouse have any significant side effects? And that is another really good question, right? Because causing those dramatic changes in mitochondria, especially in the brain, you'd be concerned that that might not be a good thing. And we have a fair amount of experience giving this compound to animals for even as long as like 3 months. We've given it daily, and we really don't see toxic side effects. It's kind of surprising. I think part of the reason why it's not toxic is because it has a half-life of about 10 hours. So if you give animals a dose of the drug, it peaks at about 10 hours and then it's gone by about 24 hours. So the cells experience the changes that I showed you for a little while, and then things can go back to normal. So that kind of oscillating drug level, I think, helps keep it safe. I'll also say that we designed the drug to look like a natural molecule that modulates metabolism in ways that are well tolerated. So we didn't try to be super clever and figure it out. We looked at what nature does to modulate metabolism in the ways we hope to change it. And then we made that natural molecule into something that's more like a drug, that's more stable. I see another question. They say a high-fat keto diet also helps in reducing weight. How does that affect the mitochondrial shape? Intermittent fasting with high fat diet is also used. Yeah, so this sort of Atkins diet, the high fat diet, is one way that people lose weight. That's another super interesting question. It's not something that we've looked at carefully. I mean, for a while, I thought maybe this small molecule was altering the formation or use of keto acids, but it doesn't seem like that's happening. So I think that's another great question that deserves further investigation. Olivia, were there other ones in the chat I should
There's
handle?
one last one. At what point of weight gain did the mitochondrial shape change?
That's another really good question. So these are pretty expensive experiments, and unfortunately, we have to sacrifice the mice to be able to look at the mitochondria. So we haven't done a time course seeing how long does it take for the mitochondria to become rounded, and exactly how long it takes for them to be fixed. So I don't know the answer to that question. The shortest time that mice were on a high-fat diet and we looked at their mitochondria was 8 weeks. If you just put palmitate on cells in a dish, it only takes like 3 hours and the mitochondria change shape. So I have a feeling like if I eat a super fatty meal, my mitochondria will change shape immediately. And when I do that occasionally, it's sort of like in the days when we were scavengers and we'd find a carcass and we'd eat a super rich fatty meal, it would be great. And our mitochondria would change shape. And that's fine when it happens once in a while. It's actually a good way to handle a fat load. But it's when this becomes chronic, when you're eating that fatty meal every day and your mitochondria stay like that all the time, it becomes a bad thing. So I think the chronicity of eating a high-fat diet all the time is part of what makes it so toxic.
Well, excellent. I think that's what we have time for, but thank you so much, Aimee, and UCI Alumni Association, just everyone for being here today. We really appreciate your time and the presentation.
Yes, thank you for coming to hear about our work and for your great questions.